Hello everyone, my name is Francisco Briceño, and today I'll be presenting a summary of my research papers on two significant agricultural threats, Xylella fastidiosa and potato virus Y. These two pathogens represent completely different challenges in plant pathology. One is a xylem-limited bacterium, and the other is a widespread RNA virus. Despite their biological differences, they both pose massive economic risks to global food security. I will guide you through their complex life cycles, the specific symptoms to look for in the field, and the integrated management strategies required to control them. Let's begin with our bacterial pathogen, Xylella fastidiosa. This is a gram-negative bacterium that resides exclusively in the xylem, the water-conducting tissue of the plant. Because it clogs these vessels, the disease it causes is fundamentally a systemic water failure. Crucially, because this bacterium lives in the xylem, it relies on xylem-feeding insects to move from plant to plant. The most significant of these vectors are sharpshooters and spittlebugs, particularly the glassy wing sharpshooter. These insects feed on the plant's fluids and act as efficient carriers, rapidly spreading the infection across vineyards. While it is most famous for causing Pierce's disease in grapevine, a condition that can wipe out entire vineyards, it is by no means limited to grapes. My research highlights that its host range is massive, affecting over 600 different plant species. This includes not just agricultural crops, but also landscape trees like oaks and elms, where it causes bacterial leaf scorch. This versatility makes it a threat to the entire ecosystems, not just specific farms. So how does it kill the plant? Once inside, the bacteria multiply and create a sticky biofilm. In response, the plant tries to defend itself by producing balloon-like outgrowths called tylosis to wall off the infection. Unfortunately, this backfires. The combination of biofilm and tylosis completely blocks the water flow. Visually, this results in the distinct symptoms shown here. In the top three images, you will see marginal scorching, where the leaf edges turn yellow or red and then die. But the most specific symptom, the one that really identifies Pierce's disease, is the matchstick symptom. As you can see in the image on the bottom left, the leaf blade falls off, but the petiole remains attached to the cane, looking like a matchstick. You might also see green islands of uneven lignification or hardening on the bark, and shriveled fruit clusters, known as raisoning. You can clearly see the symptoms on the bottom right image. Understanding the life cycle is critical for control. As shown in the diagram, the cycle begins with acquisition, where the leafhopper or sharpshooter feeds on an infected plant. The bacteria then enter the colonization phase inside the insect, forming robust biofilms within the foregut. The next step is transmission. When the vector flies to a healthy vine and feeds, it injects the pathogen directly into the xylem. Once inside the new host, we see systemic colonization. The bacteria use enzymes to degrade the pit membranes between xylem vessels, allowing them to move throughout the plant and cause systemic blockage. A fascinating biological nuance here is the vector's age. If a young nymph acquires the bacteria, it loses the infection when it molts because the foregut lining is shed. However, once an adult insect acquires the pathogen, it carries it for the rest of its life, making adult populations the most dangerous vectors. Diagnosing Xylella fastidiosa in the field can be tricky. While symptoms are useful, definitive identification requires lab work. We can use ELISA, which works similar to a pregnancy test using antibodies. Although this lab test is fast, it isn't the most reliable because it can miss low levels of bacteria. PCR is the gold standard. It amplifies DNA and provides highly reliable results, but it is less cost effective. Since there is no cure for Pierce's disease, we must focus on management. This requires an integrated approach. We can use insecticides and biological controls like parasitic wasps to reduce vector populations. Equally important are cultural practices. Aggressive weed control to remove host reservoirs, strict sanitation of pruning tools to prevent mechanical spread, and roguing or removing infected vines immediately to stop the spread. Finally, we must consider host resistance. Planting cultivars with higher tolerance to Pierce's disease is crucial. While these plants may still get infected, they hinder the systemic spread of the bacteria, acting as a buffer to protect the rest of the vineyard. The economic stakes here are incredibly high. In California alone, Pierce's disease costs the grape industry over $104 million annually in both crop losses and prevention costs. But the impact isn't limited to the US. In Europe, a strain of Xylella fastidiosa has devastated olive groves in Italy, killing thousands of trees. If left unchecked, the projected loss over the next 50 years is an estimated 5.2 billion euros. Whether it's a vineyard in Napa or an olive grove in Italy, this pathogen is an economic wrecking ball. Switching gears, let's examine our second pathogen, potato virus Y or PVY. Unlike the bacteria we just discussed, this is a potavirus, a type of RNA virus. It primarily infects the Solanaceae family, making it a nightmare for growers of potatoes, peppers, tomatoes, and tobacco. The most critical vector here is the green peach aphid, Mysis persicae. A key characteristic of PVY is that it is transmitted non-persistently. This means the aphid picks up the virus on its stylet and transmits it to a new plant in a matter of seconds. It does not need to colonize the insect's gut like Xylella fastidiosa does. PVY is notoriously complex because it exists in several strains, each causing different symptoms. The ordinary strain, PVYO, typically causes mosaic patterns and leaf mottling. However, the NTN strain, tuber necrotic, is the most feared by farmers. Why? Because it attacks the tuber itself. As you can see in these images, it causes potato tuber necrotic ring spot disease, PTNRD. These necrotic ring spots on the potato skin can extend into the flesh. While leaf symptoms like stunting reduce yield, these tuber symptoms render the crop completely unmarketable. The PVY disease cycle is a loop driven by infected plant material. The virus overwinters in infected seed tubers, volunteer potatoes left in the field, or solanaceous weeds. When spring comes, aphids begin to feed. They exhibit a behavior called taste testing, where they probe a leaf for just a few seconds to see if it's suitable. Because PVY resides on the stylet, that brief probe is all it takes. An aphid can acquire the virus from an infected weed and transmit it to a healthy potato plant almost instantly. This rapid transmission cycle is what makes the virus so explosive in the field. Accurate diagnosis is vital, and again, PCR is our best tool here because it is the only test that can reliably distinguish between the different strains, telling us if we are dealing with the dangerous NTN strain. Management is purely preventative because you cannot cure a virus. The single most effective strategy is planting certified clean seed that has been lab tested and proven virus-free. Mineral oils also help by repelling aphids. Insecticides, on the other hand, are ineffective for transmission control. The figure presented here perfectly illustrates how our management strategies must intercept the virus's transmission. As you can see in the diagram's yellow boxes, we interrupt the cycle by controlling vectors, removing reservoirs, and most importantly, starting with clean inputs. Finally, the financial impact of PVY is severe and twofold. First, we have yield reduction. Planting infected seeds can reduce overall tonnage from 30% up to 64%. Second is the quality loss. As we discussed, the necrotic ring spots make the potatoes unsellable for processing or table stock. In Idaho alone, this virus causes an estimated $34 million in losses annually. It forces growers to spend heavily on certified seed and management, cutting deeply into their profit margins. In conclusion, while Xylella fastidiosa and PVY differ biologically, they both demand our full attention. Effective management requires a combination of rigorous science, certified inputs, and strict field sanitation. In the following slides, I have included all my references for this presentation and for both research papers. Thank you for your time. I hope you found this presentation educational and informative.